Hai, Assalamualaikum warahmatullahi wabarakatuh. My name is Amanina binti Mak Musa with metric number GS57131. So today I would like to present my proposal on my PhD project. So the title of my project is to discovering the role of mating type gene in pathogenicity of Fusarium oxysporum, format specialist Cubensis tropical race 4 in Fusarium wilt. As an introduction, I would like to introduce a little bit on Fusarium wilt. So Fusarium wilt, also known as a Panama disease, that have been caused by a soil-borne fungus that was Fusarium oxysporum from a specialist Cubensis. This pathogen have been destroyed many thousands of hectares of banana plantation in tropical and also subtropical countries. So, as in history, Fusarium oxysporum Forma specialist Cubensis was first appeared in 1900s in banana plantation on Java. Next, Fusarium wilt is endemic in Cavendish type of bananas that was caused by FOC tropical race 4, was TR4, and have a biggest constraint toward the Asian countries. So they have infected the banana, and banana is actually the planting that was a very important crop and have produced with a production of over 100 million tons that was annually. And according to Malaysian Agricultural Research Development Institute, banana is ranking number two in Malaysia. That was an important crop in Malaysia and become number two also for the economy in Malaysia. So once Fusarium oxysporum infect the banana, they will in fact start at the root. Will start to colonize the root and occlude the xylem vessels, and also will cause those symptoms. That was the external wilting, leaf start to become yellowish, splitting of the pseudostem, discoloration of pseudostem, and comb. And eventually the banana will die. So actually, in managing this issue, Mardi have also highlighted some of the way on managing this issue, but eventually there is no ultimate way to manage this infection. But we can reduce, or we can prevent this to happen just by a proper hygienic practice by the farmers. For example, as follow the Mardi guidelines, the farmers have to do a crop rotation, has to make sure the apparatus used is not contaminated by the FOC spores, this become a big issue toward the towards the disease, because this disease is not easily being treated because FOC is a asexual fungus that produce asexual spores, which one of the spores type can remain in the soil for a longer period, so that's why it's not easy to treat this disease. So next is we'll move into the Fusarium oxysporum Forma specialist Cubensis tropical race 4. So TR4 is actually asexual fungus and FOC has been divided into former specialist base, and it was based on the host range, and further divided into races based on different type of banana cultivars that they infect. Next, because it is a asexual fungus, they produce three types of spores. That was microconidia, and also chlamydospores. Next, in a division of the FOC, the FOC is further subdivided. Just like I said, it's divided into races. So FOC have three races. That was race 1, race 2, and race 4. In race 4, they subdivided into two more. That was subtropical race 4 and also a tropical race 4. Subtropical race 4 and tropical race 4 is being differentiated with based on the temperature. So if the FOC remain or can remain in a low temperature, it was known as a subtropical race 4, and for a high temperature, it was known as a race 4, TR4. So in Malaysia, we should usually find TR4 strains. TR4 strains. In TR4, the range is very wide. Other than they will infect Cavendish cultivars, they also can infect others cultivars that was susceptible to race 1 and also race 2. Just I said earlier, there's no control method to manage TR4 as it will produce persistent chlamydospores that contaminate the soils for decades. So next, about mating type gene in TR4. So as I said, FOC has been known as a asexual fungus, but in a fungal biology, we when we talk about a pathogenic fungus, we always study about the sexual reproduction, because sexual reproduction is always related to their virulency. Because in sexual reproduction is we can see how the fungus mutate or birth evolution. We can see the evolution of the fungus on how they're going to manage or infect the host to make sure that they will continuously can infect the host itself, the specific host, and cause infection. So mat loci, so the definition of mating type gene is mat loci are defined genetically by segregation in progeny of elements that control sexual reproduction, development, and production of sexual spores. As in Fusarium oxysporum, their arrangement is same or similar in Gibberella fujikoroi, and here is the arrangement of the mating gene in TR4. So they in one locus of MET1, they carry three types of genes. That was MET111, MET112, and MET113 in the same locus. And as in MET111, they actually carry alpha box transcription factor. So as I said earlier, Fusarium oxysporum is actually an asexual fungi. But what is the function of the mating type gene in the FOC itself? Why they still carry the mating type gene in themselves? if actually they didn't use it. As for literature review, I put some of the related information based on the current studies about mating genes of Fusarium oxysporum. So first is pathogenic isolates of Fusarium oxysporum, cause Fusarium wilts, and have been subdivided into formal specialists, and FOC is one of the most destructive formal specialists that cause fusarium wilt toward banana plantation. That sexual reproduction is isomycete has been controlled by a single regulatory locus known as mating type, that was MET, and demonstrated that FOC carry is either MET1 or MET2, suggesting heterotalism sexual reproduction take place. However, no sexual protein structure being observed. Next is expression analysis provide evidence for a regulatory impact of encoded transcription factors are beyond specifically sexual reproduction. So as here, it's demonstrated that FOC carry either MET1 or MET2 and suggests that FOC is actually originated as a sexual reproduction fungus, but then it have been evolved and also due to adaptation to the host and make them as an asexual fungi. And also some of the studies have been done try to mate the FOC with a different strain that have a different mate gene. For example, with one FOC that carry MET1 and the other one FOC that carry MET2 gene, which make them compatible to each other, but no sexual fruiting structure have been observed. So there's no sexual reproduction have occurred in Fusarium oxysporum. Next is as a problem statement. In fungal biology, MET gene is regulatory control of sexual reproduction, and also sexual cycles is always related to the virulence of the fungi itself. Next, since identification of MET1 in TR4, its expression and function have not been investigated, as reported by a famous researcher in mating of the Fusarium oxysporum and mating in Ascomycetes fungi. That was Professor Sutomo Ari has identified that MET1 have been discovered that TR4 is carry MET1 gene, but the real function of the MET1 in TR4 is not being investigated yet. So here is my objective in order to achieve. Here is my objective throughout this whole project. So first is to determine the relative express level of MET11 in TR4 via real-time quantitative PCR to assess the involvement of MET111 in growth, conjugation, and pathogenicity of TR4 via gene deletion and mutant characterization. And lastly is to uncover the genes acting downstream of MET111 in pathogenic TR4. So here is the approach that I will use in order to achieve all the objectives that I have highlighted. First is the identification of the TR4. Second is optimization of expression level of MET111 in TR4 by qPCR. So here we will see the difference of the expression level of MET during saprophytic form of TR4 and also during their pathogenic form of TR4. Next is the production of the TR4 mutant by using split marker technique. Next we will characterize the TR4 mutants, and lastly is by using RNA sequence data analysis. First, so this is a more graphical view to see on identification and the expression level of MET1 gene in TR4. So we will isolate the TR4. We will use for the conformation. We will extract the DNA and we'll try to single digest the DNA with restriction enzyme HPYAB. Next, for the expression, to see the expression level, we will start with the RNA extraction and also proceed with the RT-PCR and also the qPCR. Next is MET111 deletion by split marker method. So actually we will delete MET111 as it carries the alpha box transcription factor and also the whole MET gene, by using a split marker method, by which we will replace the MET gene by replace the MET gene with hypromycin resistance gene from the vector that was PCV1004, and then we'll start to transform. We'll transform, and then we'll see, and then after we transform, we'll see, then we will characterize the TR4 mutants based on the morphological changes, and also we'll try on pathogenicity test. We will see how the relatedness of the MET gene toward the pathogenicity of the TR4 mutant, whether it become more virulent or also reduce their virulence. Next is the RNA sequencing. So the RNA, after we extract the RNA, we will send the RNA for the sequencing at China to see the downstream and also the upstream of the MET gene in the TR4. So we will see which genes and also which gene that will be downregulated and also upregulated, so that from there we can see we can see that MET gene is actually have effect what kind of gene, what kind of function in TR4. So as an expected result for this project is the construction of the TR4 mutant was successfully being done by using split marker approach. Next is the RNA sequencing for TR4 mutants, wild types and non-pathogenic, as we will compare the sequencing data using the wild type mutant and also the non-pathogenic of TR4 being identified and differentiated. So we want to see there's a different between these three types, TR4 mutants, wild types, and also the non-pathogenic. So there will be a difference between them. So we can conclude from that whether it's the MET gene is related to what kind of gene. So hypothesis and conclusion. Since the presence of functional MET gene does not confer sexuality in TR4, it is hypothesized that MET genes are maintained in asexual pathogenic fungus is due to its important role in virulence. So here is the reference that I've used in preparing this proposal. So I think that was last slide of my presentation, and I would like to so that's all from me. Thank you so much.